Antibody genes undergo a process called class switch recombination, during which antibody structure is partially altered. The objective of this video is to present the epigenetic basis and regulation of this process. An integral part of the body's adaptive immune system is its use of antibodies, also called immunoglobulins. Antibodies are proteins produced by B lymphocytes that bind specifically to foreign objects or antigens. An antibody consists of 4 chains, 2 identical light chains and 2 identical heavy chains. One end of each chain contains a variable region, which is developed through gene recombination and mutations to be extremely unique and specific for one antigen. The rest of each chain is a constant region that is relatively well conserved. This is the cartoon representation of an antibody, which will primarily be used throughout this presentation. An actual antibody looks more like these PyMol images. This is an anti-canine lymphoma IgG2A molecule. Here, the light chains are highlighted in red. We will not be focusing on these, or on the variable region of the heavy chains, but only the constant domains, shown here in magenta. In order to affect different functions, antibodies can exist as several different classes or isotypes. Each isotype has unique structural features, whether the number of domains it has or its glycosylation pattern. Furthermore, some antibodies naturally form polymers, such as IgA and IgM, but during class switch recombination, or CSR, only the constant region changes and the variable region is preserved. The first exon in the immunoglobulin heavy chain locus is the mu exon, which produces immunoglobulin M, or IgM for short. Thus, IgM is the first immunoglobulin isotype that a B cell produces. When it's time for the B cell to produce a different isotype, enzymes such as activation-induced cytidine deaminase, or AID, cleave the DNA at switch regions, indicated here by diamonds, located between each exon. They then excise a loop of DNA and rejoin the 2 strands. This allows a new downstream exon to be activated, leading to the production of a new isotype. Here, the gamma 1 exon will produce IgG1. Now, in order to understand the role epigenetics plays in CSR, a short review of the basics may be in order. Epigenetics is the study of cellular and physiological trait variations that are not caused by changes in the DNA sequence. That is, they are not genetically coded. Examples of epigenetic variations are DNA methylation and histone modifications. DNA methylation is a biochemical process where a methyl group is added to the cytosine DNA nucleotide. This can have the effect of activating or deactivating genes. Histones H2A, H2B, H3, and H4 are the major components of nucleosomes in chromatin and can be marked with specific histone-modifying enzymes at their N-terminal tails by a variety of post-translational modifications, such as methylation, acetylation, or phosphorylation. These histone tail modifications can modulate interactions between histones and DNA, as well as contacts between nucleosomes, leading to an open or closed chromatin state, and, eventually, activation or repression of transcription. AID expression itself is actually regulated by DNA methylation on the AICDA gene. AID is usually expressed at very low levels in naive, plasma, and memory B cells, but it is highly induced in B cells undergoing CSR. During B cell activation, the locus is demethylated, activating AICDA transcription. Primary signals from T cells and antigens couple with secondary signaling molecules such as interleukins, transforming growth factors, and interferons to induce class switching to IgG, IgA, or IgE by activating histone modifications at the corresponding switch or S region. Modified histones, together with DNA transcription, open the chromatin in S regions, thereby allowing for access of CSR factors like AID. Some common S region activating histone markers are H3K9, H3K27, and H4K8 acetylation, as well as H3K4 and H3K36 trimethylation. Thus, histone-modifying enzymes are induced to trigger chromatin decondensation in downstream S regions, such as by removing repressive H3K9 and H3K27 trimethylations, which must instead be acetylated. This creates an open chromatin state in these S regions, which allows for the access of the CSR machinery. Epigenetic dysregulation in B cells can cause aberrant antibody responses, which may result in autoimmunity or neoplasia. However, the reversible nature of epigenetic markers also makes them attractive targets for small molecule or biological therapeutics. A better understanding of the altered epigenetic processes in autoimmune diseases and B cell cancers may help pave the way for new treatments that could correct the stray epigenetic markers.